I'm really pleased to have this opportunity today to share with you some of the experiences I gained in the laboratory and helping customers here at Abcam troubleshoot their ICC experiments. And today's presentation is going to be an introduction to ICC Principles and Troubleshooting. So how do we define immunocytochemistry? So we would classify it as an antibody staining technique that's used to study protein expression and localization in culture cells. It's very widely used in basic and clinical research now. Many of our customers are using this. Researchers use cell lines in ICC, or many use primary cells as well. Usually in ICC, researchers use fluorescent conjugates for detection. So today I'm going to be focusing mainly on immunofluorescence. Through today's presentation, I'm going to be talking about the following things. So we're going to talk about selecting the correct cell line and culturing the cells, which is very important to make sure that you have a good sample. We're going to take a step-by-step look at the ICC-IF procedure itself. We're going to take a brief look at how fluorescence works. We're going to take a look at some of the results, examples showing different cellular localization, because this is one of the main advantages of immunocytochemistry. Going to take a look at some considerations to take into account when you're doing double immunostaining. And then we'll take a look at some troubleshooting. And within that, I'm going to include some considerations for recombinant protein detection. And then I'm going to hand over to my colleague Augustine, who will take you through some of our protocol resources. So to begin with, what's the main advantage of immunocytochemistry? As I mentioned, the main advantage is that you can visualize the precise cellular localization of your target, and even co-localization if you're doing double staining or multiple target staining. The advantage is that cell line samples are often easier to access than tissue samples, so it's easier to get your cell line to stain rather than waiting for some tissue. You can also test the effects of drugs such as mutagens, inhibitors and stimulators on the protein expression and localization in the cell culture. And you can of course assess the recombinant protein expression and localization. So let's start with selecting the correct cell line and culturing the cells. So to begin with, you need to ensure that you have a good positive control cell line that you know expresses the protein. And the way to do this is you can either do a literature search, or you can take a look on the manufacturer's data sheet to see if they've used the antibody in another cell line that's been successful. When you're actually culturing the cells, there's several things you need to take into consideration. You need to ensure that you're using the correct culture media, and this information you can find again on the manufacturer's instructions or the supplier's instructions. They'll be able to tell you which is the correct culture media to use. You may also find that the cells require some sort of stimulant, mutagen, or some sort of other agent to induce the expression of the protein that you're interested in. Or perhaps you may want to study the inhibition of your protein by some agents. So you need to consider what concentration do you need to use this agent at, or you may need to try a range. Again, you could start by a literature search to find out if there's any information to start with. In preparing the cells, you can grow your cells on sterile cover slips in a 6-well culture dish, or there are chamber slides available, which make staining of the cells on the slide very easy afterwards. The alternative to this is for suspension cells, where you would need to culture them as normal, and then you can cytospin the cells onto some slides. You culture the cells to the desired density. Usually this is about 70 to 80% confluent, and this usually takes about 24 to 48 hours. You would then wash the cells carefully 2 or 3 times with PBS. You can then fix the cells and go on to your antibody staining. When do you know when your cells are ready to stain? So this depends on several factors. So although we say that the cells should be about 70 to 80% confluent, this does very much depend on the target. Some cells need to be really very confluent before they start expressing the protein you're looking for in the correct localization. So you may find that this needs to be optimized. If your cells are underconfluent, they may not have expressed enough protein yet. For example, some membrane proteins, particularly cell junction proteins, they may not be expressed in the correct localization because they're underconfluent. You may have to wait till they really become quite confluent before you can see them. If the cells are overconfluent, you may have some dead and dying cells in the culture, and this could affect your staining results. Also, proteins can degrade and change localization, dying cells if they're necrotic or apoptotic. So you need to take this into consideration. So as I was mentioning, you may need to grow your cells on a matrix, for example, on collagen 1, to induce the expression of the protein you're looking for in the correct localization, and we would suggest to check the literature to make sure that this is or isn't required. Moving on, we're going to take a further look here at inducing or inhibiting your protein expression. So if you want to induce or inhibit your protein expression, there are various things that may need to be optimized. You will need to give time for the induction or inhibition to work, and you will need to optimize the amount of inducing component as well. We have an example image here. So on the right, you can see some HeLa cells that have been treated with staurosporin to induce apoptosis, and you can see here staining of caspase 3, which is activated during apoptosis. Compared to the control image on the left-hand side, you can see that the control has very little caspase 3. So you really can see the effect of the staurosporin and induction of apoptosis on the caspase 3 expression. Next, we're going to take a look at the ICC-IF procedure itself. So to take you through the procedure briefly, first of all you would need to fix the cells. You may then need to permeabilize your cells for cytoplasmic or nuclear proteins. You'll need a blocking step, a primary antibody incubation, a secondary antibody incubation, and then of course you will need to mount and store your slides, and you may like to nuclear counterstain. So let's begin with fixation. Fixation is required to preserve the cells, to preserve their morphology and their composition, and to keep the target in a good condition and at the correct localization. There's two main ways in which you can do this. You can do paraformaldehyde cross-linking fixation, or you can use organic solvents. For PFA fixation, we would recommend to fix in 4% PFA for 10 minutes only. This is quite a short fixation time, but there may still be some cross-linking of proteins, so it's not suitable for all epitopes. For organic solvents, such as acetone, methanol and ethanol, you need to use these at ice cold temperature, again for 5 to 10 minutes only, and you need to consider that they can affect the structure of proteins and affect the epitope of the antibody, and they will also permeabilize the cell. So we wouldn't recommend to use them for membrane protein detection in normal circumstances. So once you've fixed the cells, you may want to store them for a while before you continue with the staining. So in order to do this, you would need to dry the cover slip and the cells completely, place them in a slide box or wrap them gently in foil so that they're protected from the light, keep them at 4 degrees. We would recommend to keep them up to 2 to 4 weeks, not really any longer than this, before the staining. And then when you're ready to stain, you can rehydrate the samples in PBS before proceeding with the protocol. And for that, you'd need to start with the blocking step. So the blocking step is there in order to minimize the background staining from any protein-antibody interactions, and these are not specific to antibody-antigen binding. So any non-specific binding you're trying to eliminate. There are various blocking agents you can use. There's serum, BSA, milk, casein, gelatin. There are some others as well. And we would recommend to use 5 to 10% blocking agent. For ICC, it's usually serum or BSA that's used, and we would recommend to use the same blocking agent throughout the entire experiment and not to change between them. You can dilute the blocking agent in PBS or TBS, and blocking can be from 20 minutes to 1 hour, and again, you may find that requires some optimization. After this step, you can wash the cells 3 times for 5 minutes in PBS containing 0.2% tween detergent. Next, if you require it, you would need to do some permeabilization. The permeabilization is only needed for nuclear and cytoplasmic proteins. Sometimes you may need to consider it for membrane proteins that have an epitope on the cytoplasmic side of the membrane. And if you need more information, you can contact our scientific support team if you need more information about the particular antibody that you're using. There's a couple of methods for permeabilization. You can fix in organic solvent, and this will permeabilize the cells. Or you could use 0.1 to 0.2% Triton detergent for 10 minutes. Triton is quite a strong detergent, so we wouldn't recommend it to use it for much longer than 10 minutes. You could then continue to use a gentle detergent like tween in the rest of the buffers, such as the blocking buffer or the antibody dilution buffers, and this will just help keep the cells permeabilized, and when you're washing, it also helps to wash away any excess antibody. So that's permeabilization. And then you're ready for the primary antibody incubation. So you would dilute your antibody in PBS or TBS buffer containing 0.2% tween. These are known as TBST or TBSD. You can look recipes for these up on the internet. We would recommend to incubate overnight at 4 degrees. 1 to 2 hours at room temperature does very often work, but usually the staining can be more specific if you incubate overnight at 4 degrees. The optimal concentration of the antibody will need to be determined. There will be some recommendations on manufacturer data sheets, but you may find that you need further optimization from there. And the aim is really of that optimization is really to contain the most contrast between the specific signal and any background staining that you have. And use of a positive and negative control can really help there. After this step, again, you'd wash the cells. Sometimes primary antibodies are already conjugated, in which case you won't need a secondary antibody. But if you do need to conjugate a secondary antibody, then this would be your next step. So the secondary antibody would probably be conjugated to a fluorochrome, and we have many of these available. There's many on the market now, and the most common, for example, are FITC, picoerythrin, PE, Texas Red, and there are many others as well. As with the primary antibody, we would recommend to dilute these in PBS or TBS containing tween, and perhaps a blocking agent if necessary. This incubation can be from 30 minutes to 1 hour, possibly longer. Again, this will need to be optimized, and it can be done at room temperature. So again, your dilution will also need to be optimized. After this step, again another wash. So once you've finished your staining, you'll then want to mount the slides. So covering the cells with the glass on the slide will help preserve the sample, keep the cells safe, and it protects, and it also improves the optics when you're looking at it through the microscope. If you are using immunofluorescence, there are some special mountants containing fluorescent antifades available, and these help protect against photo bleaching for when you're storing the slides as well as for when you're viewing them. Many of these special mountants already contain nuclear counterstains such as PI and DAPI. We have many of these at ABCAM. For example, we have FluoroShield and BrightMount, And if you want to try some, I can recommend looking these up on the website. So once you've mounted your slides, we'd recommend store them at 4 degrees in the dark to protect from photobleaching. The fluorescence may last up to 2 to 3 weeks or longer, but for good results, we would say to take a look at them as soon as possible under the microscope. And always be aware of any photobleaching while you're observing and imaging the samples. Before we leave the procedure, I just wanted to mention an unusual tip to you, and that is to use antigen retrieval, which can sometimes be beneficial. Now, antigen retrieval is most commonly used, of course, in immunohistochemistry on paraffin-embedded sections, and the antigen retrieval is used to break the cross-links that are placed there by the paraformaldehyde, and this means that the epitope becomes more accessible to the antibody. So it's not a technique or part of the procedure that's usually used in immunocytochemistry. However, from some data we have, we know that it can sometimes be necessary to get the optimal results. So we would say that if other optimizations have been tried and the results still aren't satisfactory, particularly if you're not getting any staining, then you can consider including this step. One we tried in some of our laboratories are to apply heat at 95 degrees in 100 millimolar tris with 5% urea, and that had a pH of 9.5, and keep at that temperature for 10 minutes. This means that the proteins are then sufficiently denatured and the antibody binding sites are then more accessible. You can also check information provided on the data sheet, so very often if we're aware that antigen retrieval will help, then this information will be on our data sheets. For example, we have ATP5F1 antibody, an example shown here. So we have staining of HDFN cells. And you can see that without antigen retrieval in the image on the left, you can see very little red specific staining. And if we try antigen retrieval, obviously this seems to improve the staining and the antibody has more access to the epitope and improves the results. Next, I want to talk briefly about immunofluorescence. How does this work? So there are many fluorochromes available, as I've already mentioned. Each one of those will have its own unique spectra for absorption and emission. I've got a couple of examples here for PE and for Psi5. The excitation wavelength is the signature wavelength at which the molecule is excited. So this is the wavelength at which you shine the laser. The emission wavelength is the signature wavelength at which that fluorochrome emits a photon. So that's the wavelength at which you will detect. So how does that work? So if you have a laser at that specific excitation wavelength, it will hit an electron that's in resting state within the fluorochrome. That electron absorbs the energy from that photon and jumps up to a higher energy level state. It can't keep up that energy level state, and eventually, and quite quickly, that electron will lose the energy and drop back down to its resting state. In doing that, it releases any excess energy in the form of a photon, and that is fluorescence. That is what you are detecting. So I'd like to show you some examples now. As I mentioned before, the great advantage of ICC is that you can get very good contrast and resolution within your image, and it's really easy to see good cellular localization of your protein. So here we have some examples. So on the top we have some cytokeratin staining of bovine epithelial cells. And we can see the cytoskeleton stained here, particularly in the image on the left. You can see how well that's stained, and it's very clear. The next image down, we've got tubulin staining, which shows up the microtubules in meiotically dividing cells. Next we have some nuclear examples. So the top image here is from OCT4 staining. OCT4 is well known to be a nuclear protein. And you can see the green nuclear staining there quite clearly. More specific than that, Ki67, which is a proliferation marker, a cell proliferation marker, is known to be within the actual nucleolar itself. So you can see here the specific staining in the nucleoli. The next image, we have some more nuclear examples. We have an antibody for detection of nuclear pore complex protein, and you can see really clearly that around the nuclear here we've got green staining of this protein. The lower image is from histone H3 phospho F10, and this was kindly provided by Professor Brian Turner from the University of Birmingham, and beautifully shows the chromosomes during mitosis. And all that's doing is picking up this histone, this phosphorylated histone on the chromosomes during mitosis. This fantastic image. Next, a couple of membrane examples. And here we have two membrane junction proteins. So at the top we have claudin 5. You can see the lovely green staining of the membrane. And at the bottom we have a pan-cadherin antibody. Here we have some staining of some organelles. So at the top we have staining of lysosomes. The LAMP1 protein is a good lysosome marker, and you can see the punctate speckle staining of the lysosomes within this cell. The next one down is staining of mitochondria with complex 1 protein. And you can see the green staining of the mitochondria through the cells. Last couple of examples here. We have some lovely cardiac myosin filament staining at the top. And then we have the next one down is 58 kilodalton Golgi protein. So in the green staining in this image, you can really see the Golgi lit up green around the side of the nucleus. So that's some examples. I'd like now to talk briefly about double immunostaining because many of our customers use this technique. So what are the advantages of double immunostaining when you have fluorescence? As I said before, fluorescence detection gives really good resolution and contrast, so it really lends itself to multiple stains within a single cell line. And you can observe the localization and co-localization of 2 to 3 or more targets. And really you're just limited by the fluorochromes that you can use and the fluorescent microscope that you have. So at the bottom here, there's a couple of images that you've already seen, just to show again. So on the left we have OCT4 staining in green and actin in red. And then we have the cytokeratin image here, where the cytokeratin is in green and the actin is in red. And you really can see the difference in localization of those proteins in this way. So when you're doing double immunostaining, what sort of things do you need to take into consideration? So for each of the target proteins, you need to use a fluorochrome which emits at different wavelengths so that they're distinguishable from each other. It's good to use conjugated primary antibodies if possible. If not, you can use conjugated secondary antibodies, in which case you'll need to select primary antibodies from different host species. Although you can use different isotype subtypes, for example, IgG1, IgG2A. And again, this will ensure that different target proteins are distinguishable. You'll need to optimize the procedure for each primary antibody individually before you put them together, and we really would recommend to do this just to make sure that the reagents are working well and that the procedure is working well. And now the last section, I'd just like to take you through some troubleshooting. So there's 3 main problems you can get in ICC. Perhaps you might have no signal at all in your sample. You might have high background with lots of fluorescence. Or you may perhaps have an incorrect signal, so you've got in your cell, but it's not at the localization that you're expecting. So what can we do about these? So if you have no signal, It indicates to start with the need to optimize the protocol. You might have a defective antibody, or there may be a lack of target in the sample, and you could check that with a literature search to see how much target should be there. So the type of things that we would recommend are to increase the antibody concentration and to make sure you incubate overnight at 4 degrees. So increasing that concentration should mean that you increase the amount of staining there. Remember to permeabilize if it's a cytoplasmic or nuclear protein. Don't let the cells dry out during the antibody staining. This can give patchy staining, so some areas might have no staining, whereas other areas have too much staining. Make sure you've had the correct culture conditions of the cells for expression of the protein, and again, you can check the literature for this. It's always good to include a known endogenous positive control, and this again will indicate if your procedure is working well. Ensure the correct secondary antibody is being used, and ask yourself if it's working well with other primary antibodies, just to make sure it's working well for you. So we move on to high background. So if you have high background, it's usually from excess of antibody or autofluorescence maybe, or from non-specific binding. So for this, you need to do the opposite in a way. So you need to reduce the antibody concentration. Make sure you're incubating overnight at 4 degrees. Ensure that you're including wash steps. Wash 3 to 4 times for 5 minutes at each wash step. And make sure you include a mild detergent like 0.2% tween in the wash buffer and antibody dilution buffer. Again, you want to check that you have the correct culture conditions and that the cells were not overconfluent. Ensure there's permeabilization if it's cytoplasmic or nuclear target. That sounds quite counterintuitive, but I have found from my own experience and experience of our customers that if permeabilization isn't done when it's required, perhaps for a nuclear target, for example, that you don't get no staining. You may actually get a lot of excess staining for some reason. So just make sure you permeabilize if you need to. Again, you could check the secondary antibody, make sure that's not binding non-specifically by including a no primary control. Sometimes aggregates of the conjugated secondary antibody can give a sort of speckled appearance within the sample, so you can mix the antibody well before use. If you want to, you could even filter the antibody solution before you use it if necessary. Some cells are quite autofluorescent. You may want to check the literature for this, but they may also be autofluorescent only at a certain wavelength. So if you choose a different conjugate and view it at a different wavelength, you may find that you correct that problem. Some cell culture matrix components and proteins, again, can be quite autofluorescent. And again, perhaps you could change the conjugate that you're using to view at a different wavelength. And the next problem is if you have an incorrect signal. And this really is the most difficult one to troubleshoot, because usually it shows a lack of specificity for the protein of interest. It can't always be solved by protocol modifications. However, there are some things that you can consider. You can ensure that permeabilization, if it's a cytoplasmic or nuclear target. You can check the literature for further details on the localization in that particular cell type you're using. Check if the culture conditions are correct for the expression and for the correct localization of the protein. Are any other special conditions required? Check the secondary antibody again with a no primary control, just to make sure that's not binding non-specifically. If you want to take it a step further, you could check your sample with a Western blot and check that the band observed is at the correct size for your protein. The last piece of troubleshooting I wanted to look at was considerations for recombinant protein detection. Many of our customers are actually detecting recombinant proteins, and there are some inherent difficulties with detection of recombinant proteins that are worth considering before you start your experiments. It's good to confirm if the protein that you transfected is actually full length and to make sure it includes the immunogen sequence. And if you're not sure of this, if you're perhaps not sure of the immunogen sequence, then you can contact our scientific support team and we'll be able to help you with that. you can contact us at technical@abcam.com. Also, many recombinant proteins are tagged. Sometimes a tag can prevent the access of the antibody to the epitope. So we would recommend to place a tag on the C or N terminus just to avoid disruption of epitopes as much as possible. You may want to assess the stability of transfection by other methods, for example by RT-PCR. You may find that your transfection method requires more optimization, so it's worth going through that again. And it's very important to include an endogenous positive control sample and an endogenous negative control. That really will show you whether it's the transfection that's the problem. And if you want examples of positive and negative controls, you can have a look at individual antibody data sheets for some examples. So that concludes my presentation. Just to confirm some of the things we've gone through today, ICC-IF has many advantages. It's a fantastic technique for looking at the localization of cells. And you can also check the effect of any treatments on the cells. Very useful for the detection of co-localization of different targets because of the high resolution. There are many fluorochromes available now, which make it a very flexible technique for double staining. Useful for the detection of recombinant proteins, which are usually expressed in cell culture. The important things are to ensure the correct culture conditions and protocol optimization, and these really are the key to success. And always remember to include suitable endogenous positive and negative control cell lines. I hope that's been useful to you. I'm sure your questions have been coming in through the presentation, and I'll be taking a look at those now, and we'll be able to answer some of those shortly. Those we don't get around to, our scientific support team will be able to get back to you within the next few days with an answer. I'm going to pass you back over to Augustine now, who's going to take you through some of our protocol resources.
Thank you, Kate, for an interesting and informative presentation. I'm now going to give a brief overview of some of the products and resources that will be useful for your research. I'd like to start with introducing our CytoPainter range of products, which are an ideal tool for quick and vibrant subcellular staining. CytoPainter kits contain all components you'd need to stain subcellular structures such as actin filaments, mitochondria, or lysosomes. Actin filaments can be easily detected on fixed cells or tissues. In the image shown, you can see the labeled actin filaments in red on HeLa cells fixed with formaldehyde. If you're interested in staining mitochondrial or lysosomal structures, we also have the right tool for this. Our mitochondrial and lysosomal dyes will stain mitochondria and lysosomes respectively in live cells. In the image shown, you can see the cellular mitochondria in green. If you want to know more about our CytoPainter range of products, please visit the URL shown. Our fluorescent imaging dyes are another range of products you'll find useful in your research. Our range of imaging dyes includes DRAC5, DRAC7, and Cytrac Orange. DRAC5 is a small molecule dye which labels live or fixed cells. It's a far red dye that only labels double-stranded nuclear DNA, eliminating the need for RNAs. This makes it a rapid alternative to DAPI or hoist as a counterstain for shorter experiments. In the image shown, HeLa cells were stained with lamin B1 and alpha-tubulin antibody. DRAC5 was added to the secondary antibody mixture to label nuclear DNA. DRAC7 is a small molecule dye that only labels dead and permeabilized cells. And due to better spectral properties, it's an ideal alternative to propidium iodide. In particular, it's a useful tool for cytotoxicity and apoptosis assays. Cytrac Orange is a dye that stains both the nucleus and cytoplasm in live or fixed cells, and can be used for cell segmentation in high-content screening and exploring cell signaling and morphology. All of these biological dyes can be used in a number of different applications, and in addition to offering low photobleaching, they're compatible with other labels, so you can use them for multicolor analysis. You can find more information about these dyes on the resource page shown. Abcam's catalog includes over 2,500 secondary antibodies conjugated to biotin, enzymes, and different fluorophores, which would be useful for your research. If cross-reactivity of the secondary antibodies with endogenous immunoglobulins is a concern, for example, when the sample tissue is extremely rich in immunoglobulins, we recommend using one of our pre-absorbed antibodies, which are available as pre-absorbed against up to 3 or 8 species. Should specificity of your secondary antibody be an issue, we recommend using one of our FC-specific antibodies. In contrast to secondary antibodies recognizing the heavy and light chain, FC secondaries only bind to the heavy chain of the primary antibody. For example, a goat anti-mouse IgG heavy and light chain antibody could potentially bind the light chains of other mouse immunoglobulins than IgG. This risk is abolished when using a goat anti-mouse IgG FC secondary. All of the pre-absorbed secondaries and different secondary antibody formats are available with a number of different conjugates as shown. To help you finding the right secondary, you can use the resource shown to help you select the right secondary. If you have any scientific or technical queries, our knowledgeable scientific support team would be very happy to assist. Our support service is available in a range of languages, including Chinese, Japanese, French, Spanish, and German. For customers in the Americas, please contact our US team. For Europe, contact our UK team. And for China and Japan, please contact our Hong Kong or Japanese teams respectively. Before I hand you back to Kate for any questions you might have, I'd like to thank you for your time and wish you all the best with your research.
Thank you, Augustine. So I've had a look at a few of the questions that have come in while the presentation has been, which has been given, and I'd like to go through a few of those with you now. So Petra has contacted us and asked if there are any differences between PBS or TBS for doing ICC-IF and how to choose between one or the other. PBS phosphate buffer saline is usually absolutely fine for immunocytochemistry. TBS has a higher salt concentration and gives a more stringent wash, so perhaps if you're having problems with any high background, you may want to change to TBS. Laura has contacted us and said, can you incubate with your secondary antibody for too long? Yes, of course this is possible, and you would need to optimize the length of time that you incubate with your secondary antibody. We would recommend start with 30 minutes to 1 hour. If you incubate too long, you might start getting some background, but also, of course, what is important is the concentration and the dilution that you're using the antibody at. Jose has asked if it's necessary to do the non-specific blockage for all antibodies. We would recommend to include a blocking step. It can sometimes be possible to omit the blocking step and still get good results, but we would recommend to still include that. Jerry has asked if the fixed slides can be stored in 70% alcohol at room temperature indefinitely. To be honest, I wouldn't recommend to do this. Once you fix the cells, it's better to dry them and then keep them stored in the dark at 4 degrees. Tatiana has contacted us and said, if I fix the cells with organic solvent, I don't need any permeabilization. Is this correct? Yes, that is correct. Usually you don't need any further permeabilization if you've used an organic solvent. Just a couple more questions for you here. So from Anishka we have a cell line growing in suspension and would like to use ICC for detection of insulin receptors in these cells, how many cells do you recommend to spin onto each slide, and at what speed? So we would recommend to start with cell suspension of not more than about 5 × 10⁵ cells per mil. You can check this with the cell count, and you can dilute the cells with culture media if you need to. For most cytospin models, you would place about 200 microliters of cell suspension into each cytospin and spin at about 800 RPM for 3 minutes. Cell number for cytospinning will probably need to be optimized because different cells are different sizes, and I'd also check the instructions that come with the cytospin that you have for more information. Ahmed has asked, I have NIH3T3 cells from mouse, and the only primary we can find for detection of our target protein is the mouse primary. Would this work? Generally, cross-reactivity of an anti-mouse secondary with Fc receptors and antibody within the cell culture is not something to be too concerned about in ICC, but it does very much depend on the cell line that you're using. So, for example, Fc receptors on macrophages or plasma cells, B cell lines, that make it quite difficult. But I'm not aware that there would be any expected difficulties when these particular cells, NIH3T3, because they're fibroblasts. However, if you do get a high background, then you could consider trying mouse-on-mouse staining kits. These just include an extra blocking procedure to reduce that cross-reactivity. We have several of these available, actually. One example is the 127055. That's AB127055, and there are others as well. There's 2 other options here. You could try incubating the sections with an unconjugated FAB fragment anti-mouse IgG antibody. For example, incubate at 1 hour or overnight, and that will block your Fc receptors. Another option is to directly link your primary antibody to a conjugate in this case, so that you don't have to worry about that secondary antibody cross-reactivity. We have a lot of conjugation kits available now. They're called Easy Link conjugation kits if you wanted to look them up.